Hello everyone, my name is Niki, and I am a molecular biologist. Welcome to the first episode of this channel, where we delve into the fascinating world of science. In today's video, we will explore the remarkable process of in vitro fertilization, commonly known as IVF. This groundbreaking procedure has helped numerous couples worldwide overcome infertility challenges. Did you know that approximately 17.5% of the adult population, which is roughly 1 in 6 individuals, experience infertility, as reported by the World Health Organization? Despite this concerning statistic, there is no need to lose hope because IVF presents a ray of light in the face of infertility challenges. So, what does IVF mean? IVF stands for in vitro fertilization, and it is the process of fertilization where an egg is combined with sperm in the laboratory, outside of the female body. This procedure involves a combination of medication, ultrasound guidance, and specialized laboratory techniques. But first, let's take a deeper look at the crucial elements involved in IVF: the sperm and the oocyte, often known as the egg. Male reproductive cells are called sperm, whereas female reproductive cells are called oocytes. Each is essential to the fertilization process. Sperm are microscopic cells produced in the testes. They have a distinct morphology consisting of a head, midsection, and tail. The head contains the genetic material, DNA, and is covered by a cap-like structure called the acrosome. The acrosome contains enzymes that aid in the penetration of the oocyte's outer layer. The midsection of the sperm contains mitochondria, which provide the energy needed for movement. The sperm moves ahead toward the oocyte thanks to its lengthy tail. Oocytes, on the other hand, are the most massive cells in the human body. They are produced in the ovaries and released during ovulation. An optimal oocyte is characterized by a spherical shape, a uniform and intact zona pellucida, a translucent cytoplasm without any visible abnormalities, and the presence of a properly sized polar body. To fertilize an egg, a healthy sperm must pass through the female reproductive channel and penetrate the zona pellucida surrounding the oocyte. This process is known as sperm-egg binding. In the IVF process, the natural fertilization step takes place in the laboratory. Mature oocytes are collected from the woman's ovaries through a minor surgical procedure called egg retrieval. This procedure involves the gentle extraction of oocytes from the woman's ovaries under ultrasound guidance. To ensure the patient's comfort and security, it is often carried out under anesthesia. The expert medical staff delicately aspirates the follicles hosting the oocytes using a specialized needle. This procedure allows for the retrieval of multiple oocytes, increasing the chances of a successful IVF cycle. As soon as possible, the retrieved oocytes are taken to the lab for further processing before being mixed with sperm to begin the fertilization process. Now about the collection of sperm, masturbation is the most common way for obtaining sperm. The male partner is provided with a sterile container and a private room where they can collect the sample. Alternative techniques can be used in some situations if the male partner is unable to produce a semen sample through masturbation. These will be covered in another episode. Once both sperm and oocytes are obtained, it's time to select from the two main methods: conventional IVF and intracytoplasmic sperm injection, ICSI. Let's explore each of these methods in detail. In conventional IVF, the sperm and oocytes are then combined in a laboratory dish, creating an environment that facilitates natural fertilization to take place. ICSI, on the other hand, is used when there are specific concerns regarding the quality or quantity of sperm. ICSI involves choosing a single sperm and using a microneedle to inject it directly into the oocyte's cytoplasm. This precise and targeted approach increases the chances of successful fertilization. This approach is very effective for couples coping with male factor infertility because it can overcome difficulties such as low sperm count, poor motility, or abnormal morphology. Once fertilization occurs, the resulting embryos are closely monitored in the laboratory for a few days to ensure their proper development. The most viable embryos are chosen for transfer after being rated according to quality. The last step of the IVF procedure is embryo transfer. A tiny catheter is used to insert the chosen embryos into the woman's uterus. After the embryos are transferred, the woman will need to wait for a period of time before taking a pregnancy test to determine if the IVF process was successful. And that's it. The IVF process may be a difficult and emotional journey, but it has helped many couples achieve their dream of having a baby. I hope that this explanation has helped to demystify the general idea of the IVF process. Remember, on this journey, you are not alone. Stay tuned for our upcoming videos where we will delve into a range of assisted reproductive techniques and explore the latest advancements in the field. Thank you for watching.